trained on genomics. So the modern subject, which everybody is aware now. I'm sure you all will enjoy his lecture, and he's not new to many of us, especially the students of pathology, but the rest of the students who is from different discipline, they will enjoy his lecture. Dr. Animal.
Thank you, Dr. Kumar. And my dear students, I am audible? Yes. It's a very good morning. Loud. If you don't have the energy, how I will get your energy should infuse within me. So, well, actually, the talk was scheduled to start at 10, but it was almost 20 minutes late. Forget. Because just think about billions years ago, when there was no concept of time. The time starts from a big event, which is called the Big Bang. From there only the concept of time came. So 20 minutes is very little if you see that billions years. So that Big Bang, when it was there, then what happened during that very active environment that time? Different molecules, different things joined together to form a very basic biological molecule of the life, which is called a small RNA molecule. That RNA molecule is very catalytically active molecule, and they started replicating. And in addition with other different molecules, they started producing proteins and other things, and ultimately they gives rise to generation of this RNA molecule, and then a very small primitive molecule or primitive organism, that is the prokaryotics or primitive prokaryotes. So from these primitive prokaryotes, then all the life forms created in the earth. So whether if you talk on the plant or animal or everything started from that small RNA molecule. So we are actually brother of, or we are ancestor of that small RNA molecule. So we should have commonality, similarity between all of us. And among all these biological entity, we the human being are the most intelligent, civilized creature of the mother nature, and we are now trying to compete with mother nature. Mother nature tried to take billions of years to evolve us, and we are trying to crack the mystery of this mother nature by just sitting on a computer to analyze what mother nature has done. So after that thing, when human civilization develops from the river valley, we are now entering into the Silicon Valley. So when we started our civilization, our quest for life started. We started to know what is life, how the life evolved, how all the living being in the earth is related, and what is the element which carries the information and pass to the next generation. When this quest started beginning, the science subject started beginning. The early days of the science, when Charles Darwin and Mendel, they started to analyze these things, they started to answer these questions, and then the principles of heredity, theory of evolution came, and this is the era of classical genetics. Then several discoveries made. Meister first identified nucleic acid, composition of nucleic acid was identified. It was known that DNA is the genetic material which carries all the information. Extracrystallography of DNA was made, DNA structure was identified, and we came to know that in the DNA there are triplet codon which actually carries the information and translates it into protein. So in this era, that is the era of molecular genetics, what actually is happening? We are trying to understand how DNA is transcribed to RNA, that passes the information to the RNA. From the RNA, how the information passes to protein through the process of translation. So you know all those things. Just I am trying to put those things so that you can have that understanding about whatever the subject we are talking today. So this is the central dogma of life. So in this era, in the era of molecular genetics, what is actually happening? The classical genetics, the subject, the molecular genetics and the cell biology, these three different branches of science, they are trying to merge together to give rise to the birth of another era, the era of cellular molecular biology. And during that era, along with this subject, the tremendous technological advancement started beginning. The sequencing of the DNA was done, polymerase chain reaction was discovered, cloning and recombination DNA technology was discovered. So, with the advancement of this technology, now what happens? The classical genetics, molecular genetics, cell biology and the technological advancement, they merge together to give rise another era, the era of biotechnology. So, this era of biotechnology got tremendous momentum due to the discovery of DNA sequencing technology by two different groups of scientists, by Sanger and the Maxim Gilbert. So, when people started to know that what is the composition of DNA, people thought that we now able to crack the mystery of the mother nature, that we know what we are made of. And then people thought that we will now able to know the mystery of the human genome, what is within us. So, a big ambitious project of human genome sequencing launched, and before the targeted time, we could able to achieve the sequencing of the entire human genome. And the thanks to the more technological advancement, the engineering things, that we are able to know that the sequencing was now very much affordable. There are several different types of sequencing platforms are available. You will able to hear from couple of company persons like immunosequencing, Quagen sequencing, light technologies, the Spiro sequencing. So, there are different first generation sequencing, second generation sequencing methods, and then recently the single molecule real time sequencing based on only one DNA sequence. You will hear this kind of talk also, the smart sequencing, then nanopore sequencing. So, these are different platforms to generate the sequence information data. So, this sequence information data when was so much available, so much cheap, and what happens, the cost of sequencing decreases, the more sequence was generated, and that actually flooded the sequence database. So, huge number of sequence information now deposited into the database. So, we thought that we will able to crack the mystery of the mother nature if we will do the sequencing. So, lots of huge number of sequence information generated. Then the question started coming, oh, we have lot of sequencing data, then what? Who will, how to the different organism different, because when we did sequencing information, we observed that all the humans are similar, 99.99% we are similar, and in the human genome 95% of the genomic information is bogus, only 5% codes for important genes. So, if it is the situation, then how the organisms are different, and who will find out the function of all these proteins and how these sequences are related. So, this kind of questions started coming. So, how to analyze the sequence data? Sequence generation is not a problem, but how to really analyze the sequence data? Because what is this actually? Can anybody tell what is this? Yes, tell quickly. It is Egyptian hieroglyphs. So, are you able to read anything? So, for that you need some technique, some expertise to read this hieroglyph. Similarly, this is the sequence information, ATGC, only 4 letters. So, it is just like a boring hieroglyphs. So, if you do not have any tool to decode this information has no value. Okay. So, it was observed that again history repeats, that one subject, one individual, one institute is not possible to give solution to decode this information. So, again history repeats, and 4 major branch of science, biotechnology, information technology, computer science and statistics, they merge together to again give rise another subject called bioinformatics. Okay. So, this is the concept that how the subject bioinformatics evolved. So, when bioinformatics subject evolved, then the question, what is actually this bioinformatics? To know that, before we have to know what bioinformatics is not. If you understand what bioinformatics is not, then you will be able to realize what bioinformatics is. So, bioinformatics is not just using a computer to speed up biology. It is not just applying computer algorithm to biology, and it is not just an accountant of genomic data. Make it very clear that if you have a computer, if you have a software, you do a whatever thing is, it is not like that. So, what then bioinformatics? Bioinformatics is the creative use of the computer to define and solve the central biological puzzle. The computer becomes an hypothesis machine making prediction to be tested in the lab. So, only it is a hypothetical machine, it will assist you, it will guide you that how you can use the those information data to generate some knowledge. Okay. So, it is actually the marriage between the information science and the biological science. So, just like the central dogma of life, the bioinformatics subject also has a central dogma. This central dogma tells that if we have sequence similarity between two different sequences, if we have sequence similarity, then definitely there will be structural similarity. And if there is structural similarity, for example, this sequence similarity, if we have a query sequence, that means the sequence which you have generated, that is called the query sequence. So, if you have a query sequence and it is matching with some sequence, then if this matching sequence structure is known, that it is a zinc finger, for example, DNA binding protein, that zinc finger motif is there, then we can predict that query sequence as it is matching with that sequence. So, the query sequence should also have similar kind of structure. Okay. So, if there is a sequence similarity, there must be a structural similarity, and if there is a structural similarity, there must be a functional similarity. So, if this zinc finger motif is having the DNA binding activity and it is involved in transcription factor and other things, so the query sequence predicted to have the DNA binding activity, or it may also act as a transcription factor like that. Okay. So, if you have a sequence information, you can predict the function of that information. So, this is the power of the bioinformatics. Just by analyzing the sequence, we can able to know what is the function of that sequence. Clear? If it is not clear, you stop me, I can explain it more, because it is for the student centric program. So, definitely it is not just giving lecture from one side, you can stop me, ask me. So, so this is the central dogma of bioinformatics. So, bioinformatics subject, a huge subject, it is actually we can divide it into three basic components. The first component is development and implementation of tools and database, that is not our domain, that is the mainly domain of the computer scientists, mathematicians, and then development of new algorithm and statistical formulas, again computer scientists and the statistical persons. And third point is use of the tools to generate biological information and manage them. So, this is the domain which we are interested, that how we can use this sequence in, yes.
Sorry to
Yeah.
interrupt you.
Yeah.
To backslide.
Yeah.
So if the students haven't asked, I'll ask.
Hmm.
I just wanted to ask the students.
Hmm.
You should ask how much of the sequence should be similar to say to have the same function.
Yeah. Yeah, I'll be coming. So, just it is a central dogma, I will come now those complexities. Okay, definitely. So, bioinformatics. So, these are the three part, and our part is the last part, that is the using of those tools. So, now the title scheme. Till now it was only introduction part. So, now to have a test, have a to remove the fear that what I am going to talk, I have given a little bit introduction. So, this is the talk, that sequence based molecular phylogeny, and there I am not going to tell about the very complex things, just very basic concept and principles, so that you should understand what is those things. And because many times I have seen the students, even the faculties, even many people misuse, abuse different terms of bioinformatics, and after this training you should not do that thing. You should tell the correct terminology for correct thing. Okay. So, let us start. So, if you have done sequencing, many PhD students have done sequencing, they have done many advanced molecular work. So, you may come across, or you have generated different kind of sequence data. The sequence data may be small, maybe one gene sequence data, maybe relatively big, that is bigger genes, smaller genome of viruses, maybe big sequence data like the relatively bigger genomes of bacteria and other things, and whole genome, complex genome data of the eukaryotic genomes. Okay. So, these different kinds of sequence data you may have. To analyze those sequence information data, how we proceed? So, as it is student centric, so I will start from very basic, because there are many MSc students also involved. So, if you have a small sequence data, for example, the sequencing of small PCR product. So, if you do that sequencing from only one side, you generate a sequence information, you got a small sequence, that is okay. You may have so just do a PCR and do sequencing, or you have many more few reads, a little bit bigger. So, you need sequencing from two ends of the PCR to generate the sequence, and in that case you will generate this kind of sequence. So, you have to find out a region which is overlapping between the two strands, and then you have to do. So, this sequence read is in this direction. So, you have to just make it in the reverse complementary direction of the sequence, and then join with the rest of the sequence to get a full sequence. So, in that way, instead of means sequence from two different sense, you will have the sequence of yes. Is this okay now? Yeah. So, you will have only one sequence read. Now, you can do sequencing using a clone molecule. So, in this case, you can use a vector primer for the sequencing of your target genome, and when you will do the sequencing, what will happen? You will generate a sequence which will have the vector sequence also within it. So, many times I have observed that when you do sequencing, just raw data which you got, you submit it into the database. It should not be done. Before submission, you should carefully see that there should not be any vector contamination. Okay. So, there should not be any vector contamination into that. So, for that you have to remove the vector sequence from your sequence, and then this is the purified sequence which you can do analysis or any other things. Now, if it is a big sequence, large sequence data, then several such kind of fragments, sequence fragments will be generated, and several sequence data you have to do assembling of this overlapping sequence data, and ultimately join them to form this assembled sequence. Okay. So, for few such kind of things you can do manually by doing one by one sequence analysis, but if it is a whole genome, then three terminologies come. This terminology is called reads, contigs and scaffolds. Okay. So, a big sequence or a whole genome sequence generates several such kind of sequence data which called reads. So, they are the small sequence data of the big genome, they are called reads. When reads are aligned to form a complete sequence, they are called contigs, and these different contigs, when you align, you will get a scaffold. Okay. So, there are three terminologies, do not misuse or abuse these terminologies. Contigs, reads and the scaffolds are three different things. And for the whole genome sequencing, this contig assembly we cannot do it by manually. For that we need some sequence software, and these are different sequence software which is available. By these softwares you can do contig assembly and generates of the scaffolds. So, these are the actual sequence file, how it looks like. This is called chromatogram file or whole genome sequencing data. So, when this kind of data generated, then the question is what next? The next is to find out the meaning of the information hidden in the sequence. We need to compare this sequence with other sequence. Without comparing, we cannot able to know what sequence we have. So, if we want to compare them, then definitely the question is from where we will get the sequence to compare. For that, there are some sequence databases. So, from using those sequence databases, we can have the sequence of others, and then we can compare those sequence. So, what is this biological sequence database? They are the organized bodies of persistent data they have three basic components in any of these database. The components are update. You can update the database, it is not a closed database, it is a open database. So, every time, every seconds you can deposit anybody from the world, they can deposit their sequence to update the database. They must have a query component in the database, so that if you ask the database to give you a data, they can give you the data. So, there must be a query component and there must be a retrieve component, you can retrieve the data. So, this data biological databases are mainly of two types, one is called sequence database, second one is called structural database. Okay. So, structural database means here the structure of the proteins are deposited, this is called protein database. And the sequence database is again divided into two main part, one is sequence nucleotide sequence database and second one is protein sequence database. The nucleotide sequence database, there are three major players globally, one is called NCBI, EMBL and DDBJ. This is from USA, European countries and Japan, and these are again from the protein sequence database from European countries. So, they are different databases of not specific origin, but besides that you may have several specific sequence database, for example, rice sequence database, any fungal sequence genome database, like that, that you have specialized sequence database. These are general sequence database. So, these are the some short, that this is called National Center for Biotechnology Information, NCBI database, European Bioinformatics Institute database, EMBL, and this is the DDBJ DNA data bank of Japan. So, these are the three global database. Now, the question may come that if I will take a sequence from one database or I will submit my sequence to one database, then whether I have to submit my sequence to all these database or whether I have to get one particular sequence from one database, another sequence from another database? No. These three major database, they have the understanding among themselves that any sequence database, if you deposit any sequence within 24 hours, that sequence will be available, you can retrieve the sequence from any database. Similarly, you can retrieve any sequence from any database that may not be deposited into that database. So, that is the understanding. So, with respect to the submission and taking out the data, there is no problem, any database you can submit or retrieve. So, this is Swissprot, this is the PDB structural database where all the structures of the proteins are deposited. This is some specialized database like Oryza sativa database, this is some fungal sequence database, Magnapurthi. So, like that several specialized sequence databases are also there. Now, the question is how to deposit those sequences into the database if you have a sequence. For that there are several tools available in those databases. This GenBank is also called the NCBI database. So, they have two different options, one is called Bankit, one is called Sequin. If you have only one single sequence data, you have to submit through Bankit. If you have a large amount of sequence data, for example, cDNA data, EST data or something like that, you can submit it through Sequin. And for EMBL, you can submit it to Wavin Align, that is the option in the EMBL database. So, this is the short that Bankit in the NCBI or Sequin. So, you can go there and submit your sequence. And for the genome sequence deposition, you can again use this kind of means option in the NCBI to deposit the whole genome sequence. But before submission of the sequence, you should do annotation of the sequence. So, again another terminology, what do you mean by annotation? Many times you may come across with such kind of terminologies. So, annotation, it refers to the commentary or explanation of the information appended to the DNA or protein sequence stored in the database. So, one information, one explanatory note or something which can define that sequence. Without that thing, again the sequence has no meaning. So, you have to tell the database this is that sequence from this organism. So, these are the different kinds of annotation that shows from where you are you have isolated that DNA the country, the organism, the protein sequence, predicted protein structure, and so many things you can use as the annotation of that sequence, so that anybody, when they can use those sequence, they can able to know that the information already, the secondary information already attached to that particular sequence. So, this annotation is very much important. Before submission of the sequence, you must do annotation and then submit the sequence. So, you have sequenced a gene or genome organism and deposit it into the sequence database. Now, the question, what next? Next is to analyze those sequence. For that, you need the sequence. So, there are again terminologies, it is called data retrieval, okay? That is collection of data from database. Now, I will request every one of you, in future do not tell I have downloaded a sequence from database. It is I have retrieved a sequence from the database. So, what is the difference between download and retrieval? Can anybody tell? What is the difference between download and retrieval?
Basic English term itself should tell you.
That is the thing. What is the difference? Not clear? That is why you can frequently abuse this term, that download. So, for example, you have a file in the computer. You can, or if you have a file in your mail, you can download that file into your computer. Okay? But if I will tell that in that file download the page number 3, can you download the page number 3 only? You can open that file, you can copy that page number 3 and paste it into a new file. So, similarly, sequence database you cannot download. So, database is a kind of file, and the sequence which are there, they are the pages of that database. Okay? So, one page you cannot download, one page you can copy, retrieve. Okay? Clear? The retrieval and download thing is clear. So, you cannot download the sequence, you can download the database. Okay? You can retrieve the information which is present in that database.
Sir,
yes.
But when we are using blast algorithm in the NCBI,
It is now written, earlier it was written retrieve, now they have also get confused, they wrote many mails, they wrote clarify, but people are not understanding. They are only asking, and there are several mails also people got, this NCBI people got how to download the sequence. So, now they also got fed up and they changed, they also written download the sequence. It is now, but earlier days when the sequence databases were there, it was only written as retrieve sequence, but people could not able to understand what is retrieve sequence. Actually, the basic thing is that you are actually retrieving, you are not downloading. Downloading can be done, the database it can be downloaded, because that is a file, but the pages, the database means a file and pages are the information, sequence information. Okay. So, you cannot means download one page of a file. Okay. Clear? So, now onwards you should not tell I have downloaded the sequence, sir I will download the sequence and do analysis, no. Okay. So, what we need to, why we need to retrieve the sequence? To do two main things, one is genomics, and second one is proteomics. Genomics means understanding the interaction among genes, and proteomics means understanding the interaction among proteins. The genomics again can be divided into many things. The structural genomics means what is actually structural genomics. If you do the analysis, if you want to do a genome organization or ORF prediction, what are the genes are there, then designing of the primer for detection and other things. So, this comes under the structural genomic component. Second point is comparative genomics, that is ORF annotation, relationship of the variability and variability between the pathogens and their strains, molecular phylogeny. So, they comes under the comparative genomics. And then third component is functional genomics, that is prediction of motifs, blocks, domains in the protein, prediction of the functions of the protein, protein structural modeling and visualization of the 3D structure. So, these are coming under the functional genomics. And in the proteomics, the interaction of the proteins with other molecules and drug designing. So, these are the different components related to the plant pathogens and microorganisms, that why we need to retrieve the sequence to analyze these things. So, to do that thing we need to do some data mining, again another term. So, data retrieval means getting the information from the database, and second thing is data mining. Data mining means generation of the information from the data in the database. So, sequence data is there. So, now you want to generate the information from the data. For that you have to mine the data, and that is called why it is called data mining. Data mining can be done several ways. Here I am mainly today focusing only on this aspect, that molecular variability, phylogeny and evolution. Besides that, we can find out the genes, primer designing, protein structure and function, promoter identification. So, these are other different types of data mining which we can do from the sequence data, but only I will focus only on this aspect, phylogeny and evolution. So, before going to tell about the phylogeny, just a little bit relax, because continuously if I will bombard, then you will also get bored. So, tell me what is this? Anybody can identify what is this? Bullet, correct. What is this? Pistol. So, this bullet is shoot from this pistol. Okay. So, what is this? This is the hole inside the pistol, the nozzle inside the pistol. It looks like that. They have carved things. So, bullet when comes out, it comes out as a rolling thing, and when it hits these structures, these are the raised structure. So, the bullet got injured by the structure and some scar marks developed onto the bullet. Similarly, bullet also gives some scar mark to the pistol nozzle. Okay. So, when first bullet shoot up from this, so these are the kind of scar mark it generates. So, when it shoot up from this hole, first bullet, it got some scar mark into its body. Second bullet, the first scar mark was there, along with that some more scar mark also generated into the bullet. So, like that third and fourth, if you generate, so they accumulate different scar marks. And the crime investigators, what they do, they try to analyze whether there is some commonality between these scar marks, and then they tell that yes, these all bullets shoot from the same pistol, because they are having some kind of similarity. How? Because these scar marks are similar kind of scar marks. Okay. So, during this shooting one by one, the variation is created here, and but there has some link. Similarly, if you see, this is the process of evolution, the shooting, and this is the variation in the individual, but they are originated from the same. So, they must have some phylogeny, they have some relationship. Based on that relationship, people can tell that they shoot from the same bullet. Similarly, as I told from the first slide, that we originate from one single molecule. So, we must have, through the process of evolution, something which is there in the earlier days. So, that is the actually phylogenetics. So, what is phylogenetics? Phylogenetics is the science of establishing and analyzing the evolutionary relationship among the different organisms. How it is done? Analysis of the DNA sequence to help us understand the phylogeny of the organism. So, using the DNA sequence, we can understand the phylogeny. And what is the purpose? The purpose is phylogenetic relationship help us to understand the evolutionary history of an organism and how they are related to each other. So, that is why we need the molecular phylogeny, to understand the variability, to understand the evolution, and to understand the relationship. So, what we know from molecular phylogeny, whether our sequence has any homology with other sequence, how much similarity is there between our sequence and the sequence of others, and how our sequence phylogenetically related to other sequence. So, again three terminologies: homology, similarity, and phylogenetically relatedness. Okay. So, what does it mean? Whether they are same or different, because I have observed that students, when they are giving thesis seminar and other things, they very infrequently misuse the term homology and similarity and phylogeny, and all those things is a khichdi. So, all this terminology when they have coined, they must have different meaning, not that it is everything is same. So, what is homology? Can anybody tell what is homology? Homology, No worry. Huh? From the parent? Tell from the parent what? Any trait, yes. So, so this is a state of a gene or morphological character. You remember, it is a state of gene or morphological character that shares a common ancestry with different gene or morphological character. So, it is a state whether it is having ancestry with other sequence or not. For molecular sequence data, it is taken to mean that two sequences or even two characters within the sequence are descendant from a common ancestor or not. Okay. So, it is purely related to the ancestral relationship, whether that is there or not, again whether that is there or not. But what do we do? The term is frequently misused as a synonym for similar. We many times, every times we tell two sequences were 70% homologous. Okay. This is totally incorrect. Sequence shows a certain amount of similarity. From this similarity value, we can probably infer the sequence are homologous or not. Okay. So, similarity and homologous, two things are different. How different? Very good quote by Walter Fitch. What he told, homology is like pregnancy. You are either pregnant or not. A woman, can you tell a woman is 25% pregnant or 70% pregnant? Either it is she is pregnant or not pregnant. Similarly, the homology is the similar thing. It is a state, it is a qualitative character. We can tell whether two sequences are homologous or not. We cannot tell 25% homologous. If we tell 25% homologous, that is same as 25% pregnant. Okay. So, these are the terms we should clearly tell. If we are talking about homology, we will tell these two sequences are homologous or non-homologous sequence. You should not tell 70% homologous, never. We should not see, at least this batch of people, when they will give any seminar or any thesis seminar or anything, they should not tell 70% homologous. Okay. So, homology cannot be measured, only we can say whether homology is there or not, but we can measure the similarity. Homologous sequence must have similarity, but if there is similarity, we cannot say there is homology. Okay. If the sequences are homologous, then they must have similarity, but if two sequences are similar, 30% similar, they may not be homologous sequence. Okay. So, this is very clear, you should have this clear idea. Homologous sequence must have similarity, but the opposite is not true. If there is similarity, we cannot say, because if you do any analysis, you may see that your sequence may match with some human sequence, some plant sequence, something, but you cannot tell that this plant sequence and your organism sequence are homologous. Okay. So, homologous things actually have two major families, homologous gene superfamilies. One is called orthologous gene superfamily and paralogous gene superfamily. So, yes.
How much similarity should be there when we say that two are
Coming. When I have asked this question, definitely I have to answer this question also, coming one by one. So, orthologous gene and paralogous gene. Okay. So, what do you mean by orthologous gene? Anybody knows? These are very basic things. Orthologous gene? No idea.
Which have originated from different parents, but still have a
Still have?
means that they are from different organism and have some similarities.
So, for example, DNA polymerase of goat, DNA polymerase of human, they are originated from two different things, but their function is same kind of sequence, but they are found in different taxa. So, this is possible because of result of lineage transfer of the gene from one taxa to another during the process of evolution. And the paralogous gene, like hemoglobin and myoglobin, it is from this one taxa, two different gene, two different function, but they have the similar kind of sequence. So, like hemoglobin and myoglobin, they are having same thing, but one is transferring the oxygen to the blood, another one is in muscle. Okay, two different function, but originated from the same thing. So, this kind of things is a result of gene duplication. So, during the process of evolution, two things happens. One is lineage transfer of the genes or sequence, and duplication of the genes and sequence in the database, I mean in the organism. So, due to that, the variation and other things created, and you have the commonality as well as the difference between the different organisms.
Sir, what is the meaning of lineage transfer?
Lineage transfer means from one means taxa during the process of evolution, it transferred to, this is a parallel, means evolutionary pathway, it is going to another taxa during the process of evolution. So, it is, you cannot say that within 1 days or 1 generation it transferred. Okay. It is a huge, it is a millions of years is transfer from one taxa to another taxa. So,
When we say that the genes that got transferred through horizontal gene transfers, are they orthologous genes?
horizontal transfer, yes, those if it is transferred from one organism to another organism, that is, those are the genes which is transferred across the organism, and that those are the orthologous genes actually.
Sir, but initially if two genes were transferred horizontally, but due to some reasons in the different organisms, now their functions have changed, so then they cannot be considered as orthologous.
The function, how it will change? If the sequence are same, they will do the similar kind of function. That is the things. If the sequence are same, they will do the similar kind of function. Definitely, sequence same, it is not 100% similar sequence. It cannot be possible, because during the process of evolution, those mutation, those changes will definitely takes place. But if you see the broader sense, the DNA polymerase of goat and human, they must have the similar sequence, then only the polymerase function is there, otherwise it will not be there. So, some motifs will be there which is conserved across the families, across the genus. Okay. So, if we have a sequence, then our target is to find the homology. This is our main target. To find out homology, what madam has told, how to know that how much sequence similarity should be there to find out homology. So, we need to find out similarity or identity to probably infer whether homology is there or not. So, similarity or identity we have to find out, and for that again these two terminologies, similarity and identity. Sir, I do not know how we will finish by 15 minutes. So, similarity or identity, again two terminologies. For example, this is one sequence and this is another sequence. Okay. We would like to find out what is identity and what is similarity. So, if you see, these are the 3 sequence, in this 2 sequence, these are the 3 nucleotides which is exactly matching. Okay. So, this is called identical match or identity. So, if it is exactly matching, these 2 are similar match, purine to purine, pyrimidine to pyrimidine, and these 2 are mismatch. Okay. So, there are 3 kind of situation is possible if you try to analyze 2 sequence: identical sequence, similar sequence, and mismatch sequence. Okay. And how we are actually doing these things? We are doing a positional similarity or identity search, that first nucleotide is what in this first, second is what in second. So, this positional similarity or identity we are searching. Okay. And this finding out the positional similarity or identity is called alignment. Okay. Because very commonly we use this term alignment. What is alignment? Alignment is the identification of positional similarity or identity. So, when you find out this alignment, then we can tell that 2 sequence is how much similar, how much identical, how much non-identical or mismatch. And if mismatch is more, then they are not homologous definitely. If identity is more identity means there is more probability of 2 sequence being homologous. So, this alignment is the computational hypothesis which identify the positional similarity or identity between bases and amino acids, and this alignment may be of 2 type. One is called global alignment and second one is called local alignment. So, what is global alignment and local alignment? For example, if this is one sequence and another sequence is there, but in these 2 sequence there is only one stretch of sequence which is showing similarity or identity with the other sequence. The other stretch of sequence of both the things are not showing similarity or identity. So, only a stretch of sequence is showing similarity or identity. So, in that case this is called local alignment. So, local alignment try to find out the stretches of sequence which is showing more similarity or identity. The stretches of sequence where mismatch is more, it will not show. It will only show the region where it shares much more similarity or identity. But for global alignment, if you do, then it starts from first nucleotide of the first sequence and first nucleotide of the second sequence, and they do the entire alignment. There may be some gaps, there may not be all the sequences are showing similarity, but it will do a alignment based on the entire sequence stretch. So, local alignment is a very quick thing to know that in the entire sequence how much region is showing similarity. But if you really try to tell what is the percent similarity or percent identity, then you have to take entire sequence, the entire coverage. If you not take the entire coverage, you cannot tell the exact number of identity or similarity, how I am coming later. So, how to align these sequences? For example, we are trying to align these two sequences. There are two match sequence, 3 mismatch sequence, and there are 2 gaps. Okay? This kind of sequence, a very small sequence I am showing you. So, these 2 gaps means this sequence is 2 nucleotides shorter. Okay? So, how our main intention for this alignment is how we can increase the match sequence and reduce the mismatch sequence. And if there is some gaps, how cleverly we can put these gaps in such a way so that they can have this much more match sequence. So, we can do like this, these 2 gaps we can put here, and here we have increased the match as 3, mismatch 2, again the gap is 2 only. Or we can do like this, these 2 gaps we can create it here, and then you can see there are 5 match, there is no mismatch, and there is 2 gaps. Okay? So, in that way we can increase the mismatch. So, among these 3 different alignment, this alignment is much more perfect because it is reduced the mismatch. Okay? And this is the alignment, but you remember these gaps we have done. Actually, we do not know whether these gaps are actually there or not. So, you are tampering with the data, you are manipulating your data. So, if you are manipulating, you have to give some penalty. Okay? So, if you put one gap, you have to give one penalty. If you put another gap in another region, you have to give another penalty. But if you put a gap first and then you put another gap just after that, then the penalty will not that much, because already one manipulation you have done in that region, you can do another more manipulation. Okay? So, due to these things, so what actually have done? So, if this match is 2, if you give a means number or value of this alignment 2 and this alignment value as 3, this has 5, but as you have put 2 gaps here, then 2 marks will be reduced from here or 1 marks will be reduced from here. So, you have done, you have got the correct answer, but you have cheated. So, 1 marks will be deducted. Okay? So, like that some score value will be generated, and the more score value means more probability of the correct alignment. And as you are doing this kind of gap, so you have to give some penalty. So, this is called open gap penalty. When you open a gap, that is called open gap, and when you extend that gap with another gap, that is called extended gap penalty. So, if you do, anybody has done sequence alignment, you will see that it is written that how much open gap penalty was there, how much extended gap penalty was there. So, that means to do the proper alignment, you have to give some penalty, and then the correct alignment will come and the score value will come. But it is only 5 nucleotides, so many alignment is possible. If it is a huge large sequence and large number of sequence, only 2 sequence we are doing with 5 nucleotide, and so many possibilities there. And if it is 1000 nucleotide, or if it is a genome, and it is like that, if you have thousands of genome, then how to do? those analysis by manually it is possible? No. For that we need dynamic algorithm or programming. Those programming will do alignment for you and tell you the result. What result it will give? The programming generates some scores, these points, that if you some penalty and other things, alignment has how much point, that is called score. So, more score indicates more similarity, and they also generates a e value. This e value means the probability of not matching the sequence with the other sequence. So, if e value is 0.003 means the probability of not matching your sequence with the sequence which is deposited is 0.003. That means the probability of matching is 99.99 like that. Okay. So, more score means more similarity, less e value means probability of being homologous sequence is more. Okay, clear? So, there are two terms again, scores and e values. So, if the e value is less, that means there is more probability of those two sequence being homologous. Okay. So, this is the kind of score the softwares actually generates, the different value points. So, minus means there are more gaps they have created, so score is minus. Okay. So, this is a kind of blossom matrix used by the different software. So, the sequence alignment tools to do these things, what are the different softwares or programs which we use? These are the different programs: BLAST, FASTA, Bleach, Beauty, ClustalW, Mega. So, these are the different softwares or online programming which we do to do the basic alignment. Now, what is BLAST? It is not bomb blast. Yes. How do we create or software create? Yes.
So, if it is aligning in a normal fashion, so it is going to see that if two sequences are same or
Yes.
different.
Yes.
Then why are we trying to align?
Yes. Now, yes, we have these two sequence. Now, this sequence when I have done, we got in this sequence we got only 5 nucleotide. Here we got 7 nucleotide. Okay. That means the first nucleotide is 2 nucleotide shorter. Now, these 2 nucleotide, that means there is a mutation or deletion from that. Now, deletion has taken place at the last, or deletion may taken place here, or deletion may taken place here, anywhere it ultimately when sequence comes, it is continuous sequence, but during the process of evolution that 2 nucleotide deletion may take place from here also. So, it has actually it is coming like that. If the 2 nucleotide deleted from here, then only they are matching properly. That means, it may be possible during the process of evolution the gaps has created in this region, not at the end, because sequence when you read it is continuous, but actually during the process of evolution where that gaps has created, that we also do not know. That is the probability that that gap may be created here, may be created here during the process of evolution. And the more, how we will know that yes gap has created where? Because if you have more match sequence, then the probability is more that the actual during the process of evolution the gap was created here, because then only the more match is coming. Clear?
What if it has not taken like that and
Then this is the condition. If you directly think that only the gap has been created at the end, then the match is less, mismatch is more, then your inference is that these two sequences are non-homologous. Okay. So, you have to, because not that all the sequence will be in the same length, okay. There will be different size of sequence. So, if you are not able to, that means in different sequence different deletion has taken place, or in another word in other sequence different addition has taken place. So, where that addition has taken place, where the deletion has taken place, if you do not take that into consideration, your analysis will not be proper. Clear? So, sir, what I will do? I will continue because it takes a little bit. So,
These are the points which are very
No, you can
required for the
maybe
students.
That is why for the students,
Now, if you want, we can give a break for tea now.
you can get a.
We can take another half an hour, maybe up to 12 o'clock. There's
Yes.
no problem.
Yeah, yeah, that's
Because this basic information, they are doing sequence analysis, they are doing all those things without knowing the basic. Blast, sir, I have blasted the sequence. Hari blast, ye ke bomb blast hai ke blasted the sequence? Blast is the name of an analysis. I have blasted the sequence. I have heard that students are telling like that. I have blasted the sequence and got this. Blasted means they think that it is a bomb blast. So, tea break will have?
Yeah, we'll have a tea break.
Okay.
For
Yeah.
another